Hello everyone, am I audible?
Yes, ma'am.
Okay, let us begin with the live interaction session for week 4 for the course Classics in Neuroscience. Uh, Before I begin, does anyone have any doubts or any questions that you would like me to address from the course lectures? Is there anything that you would like me to address first? Okay, then I will take it that you do not have any doubts. In case you happen to have any doubts or any questions, feel free to unmute yourself and ask me or drop a question in the chat box. Let me first begin with discussing the previous year assignment questions, followed by discussing some of the key concepts that were covered in this week's session. Is my screen visible?
Yes, ma'am.
Okay. So, first question. Which physiological effect was the first example of a synaptic response, as well as the first direct recording of the effect of a synapse, according to the findings of various groups, including Gopfert and Schaefer, Feng, and Eccles and O'Connor? So, all of these groups, Goffert and Schaefer, Feng, Eccles, and O'Connor, all of these groups worked on understanding what happens at the synapse in the context of the neuromuscular junction. And they found that when the synapse of the synapse at a neuromuscular junction, basically where the nerve innervates the muscle, that particular synapse, in that case they observed that there is a phenomenon called end plate potential that is initiated in the postsynaptic side. Basically, in the muscle, for example, if the postsynaptic side is the muscle, then in that case, they observe that the action potential and at the presynapse initiates an end plate potential in the end plate regions of the postsynapse. And this end plate potential that is initiated then gives rise to an action potential in the muscle. So this is what they observed by independent experiments. So this physiological effect that they observed was called end plate potential because it initiated at the end plates of the postsynapse. Next question. Which two key advances in neurophysiology were established through collaboration with Paul Fatt during the intracellular study of synaptic transmission at the frog neuromuscular junction? So, Paul Fatt collaborated with both Bernard Katz and Jack Eccles. And these collaborations of Paul Fatt was an American. He collaborated with both Bernard Katz, who was a Jewish who was Jewish, and he had to run away from Germany. He had to run away from Germany because of the persecution by the Nazis. So Paul Fatt and Bernard Katz had initially collaborated to study the muscle end plate potential. So we just discussed what end plate potential is. I hope you understand what it means. So Paul Fatt and Bernard Katz, they collaborated and they studied the phenomenon of muscle end plate potential using the frog neuromuscular junction. Uh, then Paul Fatt also collaborated with Jack Eccles, and he to understand the the nature of synaptic inhibition. So basically, at the synapses, uh, what happens is not just excitation happens, but also inhibition can happen of the postsynaptic side. So the presynapse can give rise. Now we know that those are neurotransmitters, inhibiting neurotransmitters, but that at that point of time, people did not know about the concept of excitatory and inhibitory neurotransmitters. At that point, people were still trying to understand whether the neurotransmission that is happening is electrical in nature or chemical in nature. So they did not know about the nature of synaptic transmission. So during that time, Jack Eccles was very keen to understand about the electrical. He was a keen proponent of electrical basis of neurotransmission. So he said that the neurotransmission that happens is electrical in nature. So at that time, while Eccles was studying the spinal motor neurons, he also collaborated with Paul Fat and to study the neuromuscular junction and they uncovered that the synaptic nature of inhibition, how it happens. Next, what did Eccles focus on in his studies of synaptic transmission in sympathetic ganglia, leading to a conflict with emerging pharmacological evidence by H. Dale, Wilhelm Feldberg, Lindor Brown, and others? So initially, when Jack Eccles was a much junior person in the field, he was a student of Charles Sherrington. So we in the previous lectures and also in the current in the current week's video lectures, you would have heard that Charles Sherrington, who started his work. By working on the reflex of in using the spinal cord as his model. So and he Charles Sherrington, who also coined the term synapse. So Charles Sherrington had several students, and one of the last students of Charles Sherrington was Jack Eccles. And Jack Eccles was an Australian, so he was born and brought up in Australia as a son of Catholic school teachers, and then he went on a scholarship to England. So Jack Eccles was one of the last students of Charles Sherrington, and he also started working on the spinal cord. So Sherrington, one of the speciality of Sherrington was his work on the spinal cord, and Eccles, being his student, was also started working on the spinal cord. So his focus was studying the synaptic transmission in sympathetic ganglia, which are basically present on the sides of the spinal cord. So while he was trying to understand how this synaptic transmission worked, he interpreted his results in terms of electrical transmission. So he was a keen proponent of electrical transmission rather than chemical transmission. He was not willing to he was very strongly opposed to chemical transmission, and he strongly believed that there is electrical transmission that is happening at the synapse between the presynaptic terminal and the postsynaptic terminal. But other people, the at that time the stalwarts in the field or the more senior people in the field, like H. Dale and Wilhelm Feldberg and Lindor Brown and others, these people they had pharmacological evidence that what the transmission that is happening between stimulated nerves and the postsynaptic cells is chemical in nature. So there were very heated debates in different conferences and so on between a much junior Eccles and a much senior H.H. Dale, because their views were completely conflicting. So they used to have very heated discussions about whether the transmission is electrical in nature or chemical in nature. So, Eccles's focus on studying synaptic transmission in the sympathetic ganglia led to a conflict with these people on the basis of his idea that there is electrical transmission between stimulated nerves and postsynaptic cells. And so, I hope you have understood the answer to this question. Do you have any doubts? Does anyone have any doubts?
No, ma'am.
Okay. Next question. In the context of crayfish sensory receptors, what is the mechanism described as the clearest demonstration of the receptor potential in response to a stimulus? So, before I discuss the answer to this question, let me explain what receptor potential means, and then we can understand what is the correct answer to this question. So, uh, yeah. So, the crayfish has something called stretch receptor cells. So these stretch receptor cells basically they innervate the muscle spindle of the crayfish. And whenever there is any stretch, these receptor cells are able to sense that stretch of the muscle spindle. So these are basically sensory uh, cells. So these sensory cells, how are they? So whenever there is any stretch of the muscle spindle, what happens is there is a receptor potential that is generated in these stretch receptor cells. So upon muscle stretch, there is a depolarizing potential that is generated in these stretch receptor cells, which are called receptor potentials. And these receptor potentials that are generated that result in the initiation of an action potential in the cell, followed by transmission of the action potential to the crayfish nervous ganglia. So basically, this is the basically it explains how the the stretch of the muscle spindle is sensed by these sensory receptors, that is the stretch receptor cells, and the because of which there is a receptor potential that is generated in these stretch receptor cells, and that results in the initiation of action potential and the transmission of this potential to the nervous ganglia. Okay, now coming to this question. In the context of crayfish sensory receptors, what is the mechanism described as the clearest demonstration of the receptor potential in response to a stimulus? So, what they are basically asking is, how do you know that there is something called a receptor potential that is being generated in these sensory receptors? How do we know? Because there is a depolarizing potential that is being generated in the dendrites, and then that is being generated that spreads from the dendrites to the cell body. If a hyperpolarizing potential is initiated, that will not, that will inhibit neurotransmission, that will not activate, I mean, that will inhibit action potential, that will not activate, that will not cause the initiation of an action potential. So that is why the first option is incorrect. The second option is incorrect because first a depolarizing potential has to be generated, and that depolarizing potential is being generated, and that depolarizing potential which is being generated, it is spreading from the dendrites to the cell body. And because of the presence of this depolarizing potential, we know that there is a receptor potential that is being generated in response to the stimulus, which is basically the stretching of the muscles. And of course, like I described, the depolarizing potential has to be is being generated, and that is spreading from the dendrites to the cell body, finally causing the initiation of the action potential, and finally followed by the synaptic transmission. So it is the it is the presence of a depolarizing potential that is taking place, which demonstrates that there is a receptor potential in these sensory receptors. Next, what is recorded from the frog olfactory epithelium in response to smell stimuli, serving as a useful monitor of the overall stimulating effectiveness of different odor compounds? Just a minute. Let me just. Okay. Okay. So we are. So the recording is from the frog olfactory epithelium, and in response to smell, right? And it is it is serving as a monitor. I mean, it it is to determine the response to different odor compounds. Okay. In that regard, let us look at the options. Electroretinogram. So, electroretinogram, from the term itself, you understand electroretinogram. So, it is something that will record the fields from the retina. Electrocardiogram records fields that are generated by the heart. Electroencephalogram records fields that are generated by the brain. Electroolfactogram is that which can record fields that are generated by the olfactory epithelium. So that's why the correct answer to this question is electroolfactogram. Next, what is the primary purpose of the gemmules on dendrites as described in the discussion? So, gemmules is the term that was uh, given by Chang to refer to dendritic spines. And what Chang said is, so I have a, yeah. So what Chang said is that functionally, so according to him, this was his observation, that functionally the gemmules on the dendrites constitute a mechanical barrier preventing the synaptic knobs from reaching the stem of dendrites directly. And hence they serve as a limiting factor for synaptic excitation. They also delay and attenuate the process of synaptic excitation because of the high ohmic resistance of the gemmules' extremely slender stalks. So H.T. Chang coined the term gemmules. One second. Yeah, H.T. Chang coined the term gemmules to refer to the dendritic spines. And according to him, these gemmules or the dendritic spines were a barrier to prevent the synaptic knobs, or basically the presynaptic terminals, from reaching the dendrite stem directly. So basically, these dendritic spines attenuate the synaptic transmission because they prevent the presynaptic terminal from interacting directly with the dendritic stem itself. And how do these gemmules do that? So according to Chang, these gemmules, these dendritic spines basically have very slender stalks. So because of these slender stalks, there is a very high ohmic resistance to the uh, synaptic excitation. Because of the high ohmic resistance, there is attenuation of the signal. Next, which neurological phenomenon became a valuable tool for investigating the trisynaptic circuit in the hippocampus, leading to the exploration of long-term potentiation as a potential mechanism for the synaptic basis of Hebbian learning and memory. So what happened is there was a graduate student who actually whose experiments actually led us. So he was trying to understand the population spike. So population spike is let me go to the slides and explain. Maybe that will be better. Yeah. So there was a graduate student who was trying to understand population spikes in the hippocampus. So basically, there is a sharp wave that he observed when he was carrying out experiments in the hippocampus, and he observed that this population spike, that is the fields of the extracellular potentials of several synapses at once. So he saw that this population spike is initiated in the distal apical dendrites by sensory excitatory synapses, and then when once it is initiated in the distal apical dendrites, it propagates through the dendrites towards the cell body and the axons. So this showed that there is a uh, like the dendrites are not just passive structures, but they are also active structures because of their ability to because of the ability of these population spikes to be initiated in these dendrites by the synapses. And this phenomenon of the population spike was later, it was also led to exploration of long-term potentiation or LTP as a mechanism for the synaptic basis of Hebbian learning and memory. Next, what term did Ramoni Cajal use to describe the overall direction of the flow of activity from contacts on the dendrites and soma out to the axon and to the axon terminals in his collaboration with Alfred van Gehuchen in the early 1890s? So initially, remember, this is the early 1890s. It was much before the idea that there is something called synapse, and there is what is the flow. I mean, how, what is, people had not yet invented the I mean, people had not yet looked at the cells under the electron microscope. People did not know about the fact that dendrites have dendritic spines. Dendrites are active structures. I mean, they have active properties. None of these things were known at that time. It was much early. So in the early 1890s, when Kahal was trying to understand how the information is flowing across the within the neurons and across the neurons to other neurons and so on. In his collaboration with Alfred van Gehuchen, he came up with the idea. So Kahal was a very imaginative person. His powers of imagination were extremely huge. So Kahal described that, I mean, he did not know that dendrites were active structures, right? So what his idea was that there is some flow of information from dendrites to soma to the axon and then to the axon terminal. Right now, this seems like a very, I mean, this is a very obvious thing for us. We understand this to be a very obvious statement, but at that time, the idea that information actually flows across the neuron from these different regions and then maybe is also going from one neuron to the other was actually very revolutionary. So the term that he used to describe this direction of the flow of information from, so there is a direction that he is proposing in this, right? From dendrites to soma to axon terminal. And the term that he used for this was called the law of dynamic polarization. So the correct answer is the law of dynamic polarization. Next, what classical concept describes the backward spread of the action potential in a neuron, informing cell bodies and dendrites about the occurrence of an output and influencing future responsiveness, particularly in neurons like motor neurons with passive dendrites? So initially, people like when people were studying the action potential, they thought that the action potential is spreading forward. So it is according to them, initially they were thinking that action potential is probably being initiated in the dendrites or in the cell body itself, and it is in response to the presynaptic uh, like in response to the neurotransmission from the presynapse, there is some there is action potential that is being initiated. Probably, at that point, people thought that probably this is probably getting initiated probably in the dendrites or cell body or something, and maybe it is from there itself it is getting initiated and it is propagating along the axon and it is reaching the axon terminal is what they thought. They did not know that it is actually not the case, that the action potential is actually initiating at the axon initiation segment. That was this concept came actually later when people were trying. So when Jack Eccles, for example, Jack Eccles and his group were one of the people who were trying to understand whether this action potential is going just forward or it is going backward also, whether it is spreading backward from the axon to the cell body and dendrites and so on, or is it just going forward. This was the question that Eccles and his group tried to address. And they saw that this action potential is indeed also spreading backward from the axon to the cell body and dendrites. So this spread of the action potential backwards from the axon to the cell body and dendrites, they they went on to check, okay, this is spreading. But where is the so they did then where is this action potential actually being initiated? It is spreading from the axon to the cell body. If and it is also if the impulse is coming from the dendrites to the cell body to the axon, then also it is spreading backward to the cell body. So it is not the action potential that is going from the dendrite to the cell body to the axon terminal. It is also able to go spread backward to the cell body and the dendrites. In that case, where is the action potential being initiated? Is it being initiated in the dendrites, in the cell body, in the axon, or where? Interestingly, they found that this action potential is being initiated in the axon initiation segment, or the region that they had called the axon initiation segment. To their great surprise, that is where the action potential was being initiated, unlike what they had thought, because they had thought that the action potential is being initiated probably in the dendrites or cell body, but that was not the case. Then they realized that there is an integration of the receptor potential, or the summed potentials in the dendrites and cell bodies with the action, like that is getting converted to the action potential that is propagating along the axons. So there is an integration that is happening in the dendrites and cell bodies. So they called this as the integrative organization principle. I hope that is clear. Do you have any questions? Does anyone have any questions?
No, ma'am.
Okay. Next. What concept was born from Rahl's realization that recorded transients were slower than expected, leading to the modeling of a cell body attached to a long cylinder representing the dendritic tree? So, Wilfrid Rahl, he had been working on uh like these, so he was a he had worked with Jack Ickless, and he also worked with Archibald McIntyre. Let me just go to the presentation and show it, discuss it better. Yeah, so Wilfred Rahl was someone who had been actively working on the electrical currents in the cell bodies and dendrites. So, when he saw some results that were published by Jack Ickless about the backward spread of transients from the axon into the cell body and dendrites, he came to understand it, or he came to model it in terms of equivalent cylinders. So he said that each of the dendritic tree could be I mean, each of the dendrite sorry, the cell body and the dendritic tree, and these could be modeled in terms of the cell body being attached to a long cylinder. So the dendritic tree could be equivalent to a long cylinder. because so he observed that the transients that were being recorded, the transients that were coming back to the cell body, those were slower than was expected. And he thought that this is because the currents were going into the dendrites. And so he said that based on this realization, he said that the this could be modeled in terms of a cell body that is attached to a long cylinder, and that long cylinder can represent the dendritic tree. And this concept was called the dendritic dominance concept. But what happened was that Ral's this concept of Ral's that he tried to publish was rejected, and it was rejected by none other than Jack Eccles. So Jack Eccles said that no, these are probably some instrumentation related problems or something. That is why those the transients are slower than what is expected, and that is not what you are that is not what you're proposing. So Ral's paper was initially rejected, and this rejection impacted not just Ral's career, but also the field itself, because the acceptance of this dendritic dominance concept also got delayed because of that. But he finally managed to publish this concept, his concept of the equivalent cylinder in experimental neurology in 1959 and 1960. But there was a delay because of rejection of Ral's model by Jack Eccles himself. So the correct answer is dendritic dominance concept. Next, who coined the term synapse and helped describe its functions? Like we have already discussed several times, the term synapse was coined by Charles Sherrington or Sir Charles Sherrington. So Charles Sherrington coined this term in around 1897, if I'm not wrong, and he was the one who after several he came up with this term after like a lot of thought. He discussed with several people, and they proposed that hey, you can call this as synapse. So synapses means contact or connection. So he proposed the term called synapse to define these regions of contact or connection between individual neurons. Next, what determines the frequency of miniature end plate potential? So before that, let us first discuss what the miniature end plate potential is. Let me just go back to the slides. Okay, I think it is. Okay, it is not in the. Okay, so we discussed previously what an end plate potential is, right? We discussed that end plate potential is the potential that arises uh, at the neuromuscular junction at the postsynapse, that is basically, for example, the muscle. So at the end plate of the postsynapse of the neuromuscular junction, there is a potential called the end plate potential that gets initiated when there is synaptic transmission from the presynaptic terminal, right? This end plate potential, while Katz was trying to study this end plate potential in Hill's lab, so at that time they observed that there are these miniature, so there are these very small miniature in-plate potentials that they are able to see. That means the smallest value of the in-plate potential that they were able to see, like they were able to record. This they termed as miniature end-plate potentials. So these miniature end-plate potentials, now we know that they arise from the fusion of individual synaptic vesicles with the presynapse, and these individual presynaptic vesicles release neurotransmitters that can go and interact with the receptors on the postsynapse. At that time, they this was at the level of physiology, right? They were trying to understand what they were trying to see from the recordings at that point. Although independently, there were other the electron microscopists were trying to look at the structure of the synapse, the morphology of the synapse, and they had observed the presence of these vesicles. This observation of a miniature end-plate potential was something that that was independent of the observation of the synaptic vesicles. So when they were recording these end-plate potentials, they saw that there were these very miniature or small uh, spikes that they were able to see, or the smallest value of the end-plate potentials that they were able to see. So they came up with the idea that these miniature end-plate potentials are because of neurotransmission from the presynapse. The frequency of these miniature end-plate potentials is determined by the presynaptic terminal. So basically, how much will be the how frequent the end-plate potential will will happen is something that is determined by the presynaptic terminal, whereas the amplitude of the miniature end-plate potential will be determined by the postsynaptic terminal. This is what the observations they made were. Now we know that these when this after they observed these miniature end-plate potentials, and after they came up with this idea of the frequency being because of the presynapse and the amplitude being because of the postsynapse, when they got to know about the other independent research where the other people had seen these synaptic vesicles under the using electron microscope and so on. They put things together, and it was understood that these miniature end-plate potentials are basically those that arise because of synaptic vesicle fusion of individual vesicles. So each individual vesicle that is fusing, each individual synaptic vesicle that is fusing by exocytosis releases its uh, content, the quanta or the minimum neurotransmitter content that it has into the synaptic cleft. And these neurotransmitters, so each synaptic vesicle will have hundreds of neurotransmitter molecules, right? These neurotransmitter molecules go and bind to their postsynaptic receptors, and that gives rise to the postsynaptic end-plate potential, correct? So then they realized that, okay, this miniature end-plate potentials that they are seeing is because of the synaptic vesicle fusion. And now we know that, if with that in the context, it becomes even easier to understand that. So the frequency of the miniature end-plate potentials will depend upon the frequency of the frequency of the fusion of the synaptic vesicles with the presynaptic membrane. Because how frequently the synaptic vesicles are able to fuse with the presynaptic membrane will determine the frequency of the MEPP that is generated. Whereas the amplitude of the MEPP will depend upon the interaction of these neurotransmitters with the receptors that are present on the postsynaptic terminal. So the density of the receptors or the type of receptors that are present at the postsynaptic terminal, that will determine the amplitude of the MEPP. So now we can understand this in terms of the actual mechanism that is happening. But initially, when the scientists were uh, like they observed these MEPPs, just on the basis of their observations, they were able to conclude the fact that the frequency depends on the presynaptic terminal and the amplitude depends on the postsynaptic terminal. So just I hope you are able to appreciate the brilliance of these scientists in their ability to use their extremely the observations that they have made and be extremely critical of these observations and actually come up with such very elegant conclusions, because now at this point we are able to relate those because now we know how the neurotransmission works, the fact that there are synaptic vesicles, they fuse, they release the content, and so on. But those people did not know those things initially. Those observations were happening as these people were conducting experiments. And their ability to actually understand what is happening based on the systems level of observations that they were making was very remarkable. Next, why was aplysia considered an important model system for neuroscience? So, aplysia is the garden slug. So, this model was like brought into was extensively studied by. Does anyone know? Okay, wait, there's a question. But how does it help if the signals or currents come back to the cell body from the axon initiation site? Shouldn't it travel to the next dendrite? Okay, so Soumya, so I'm not saying that the action potential that is being generated does not propagate along the axon. Of course, it propagates along the axon. So, the action potential that is being generated at the axon initiation site, it does get we know the Hodgkin and Huxley model, right? It does propagate along the axon. The action potential does travel along the axon, and it reaches the synaptic terminals. That is true, but the action potential, when it is initiated, some of the information also comes back to the cell body and the dendrites, such that so that the cell knows that the whatever impulse was initiated in the dendrites and was integrated in the cell body has resulted in the initiation of an action potential, and it will this the information the this backward in spread from the axon to the cell body will also help in the future responses that will happen. So how the neuron responds in future to the next set of impulses that it receives, that is also influenced. It is basically like a feedback in a way. So you can imagine it as a feedback from the axon to the cell body and dendrites. So basically you can imagine it like the axon telling the cell body and the dendrites that, hey, the information that the action potential has been initiated and successfully initiated, and the information is getting is being taken forward. And this will help the neuronal cell to decide how to respond to the future impulses that it receives from other neurons. Okay, going back, what were we discussing? Okay, why was aplysia considered an important model system for neuroscience? Yeah. So Ladislav Tauc was a French scientist who worked extensively on aplysia. These are all of the these are many of a lot of a lot of the early investigations that Ladislav Tauc carried out, all using aplysia. And the reason he used aplysia was because this garden slug was had a very simple central nervous system, making it easier to identify even the individual cells across different animals. So if you're able to identify which cells you are working with, which neuronal cells you are working with, you can also identify if you how like changes that you are subjecting them to, changes that you're subjecting the organism to, are affecting these cells, right? So it makes it much easier to study what is happening at both the whatever you're doing at the level of the system or the level of the organism. You can also understand what is happening to the cellular level, correct? And you can basically also make recordings also much more easily in this organism. So that's why aplysia was a very crucial model system. In fact, uh, Ladislav Tauch's work on aplysia, when he presented this work at a conference, it also influenced Eric Kandel to to choose aplysia as his model for some of the many remarkable learning and memory related experiments that Kandel carried out. You're welcome, Soumya. Yeah. So, why was aplysia considered an important model for neuroscience? Because it had a simple central nervous system. Its central nervous system was simple, but it was still capable of complex behaviors such as reflex and so on. Next, what type of recording method did early scientists like Edgar Adrian and Zotterman use to study sensory receptors? This we have already discussed in the previous week lecture, live interaction session, that Edgar Adrian at that time had access only to the extracellular recordings. At these were early scientists. At that time, the intracellular recordings had not come into the picture. They had not yet been developed, right? So Edgar Adrian and Zotterman had only had the option of using extracellular recordings to study sensory receptors. They did not have the option of using intracellular recordings. Despite that, by just using extracellular recordings themselves, Adrian and Zotterman were able to understand how information. They were able to study sensory receptors. I think they used the frog, if I'm not wrong. Uh, they were able to study these sensory receptors but just by using extracellular recordings, because they managed to dissect two single fibers to study these sensory receptors. What is recorded? I think we have already discussed this question. What is recorded from the frog olfactory epithelium in response to smell stimuli, serving as a useful monitor of the overall stimulating effectiveness of different odor components? So the answer to that is electro olfactogram. Does anyone have any doubts regarding this question? Do you have any doubts?
No, ma'am.
Okay. Next. How does information first enter a neuron? The answer to this is very simple, right? We have already discussed that information enters a neuron first through the dendrites. Then how does it go? Can someone tell me how information passes along a neuron? What is the flow of information in a neuron? No? Okay. In that case, let me tell you. So information first enters a neuron through the dendrites, goes to the cell body, passes along the passes to the axon terminal, and then there is synaptic transmission. This was what was proposed by Ramoni Kahal, this flow of information. Later, we also understood that there is a backward spread from the axon to the cell body and the dendrites. Even later, we also understood that the dendrites are actually active. They have active properties. They are not just passive elements. They do not just transmit the impulses that are entering the neuron. They also play a role. Both the dendrites and the cell bodies play a role in integration of this information. Is that clear?
Yes, ma'am.
Okay. Next. What technological advancement helped support the neuron doctrine in the 1950s? So, like we have discussed, the idea of the idea that neurons are individual entities that interact with each other, not via connections but via contact, was something that was proposed by Ramoni Kahal, right? But during Kahal's time, there was only the light microscope that was available. There was the electron microscope came much later in the 1950s. Based on Kahal and Wilhelm, his observations, the neuron doctrine, I mean, these people and several others, the neuron theory first came. This was then elevated to the status of the neuron doctrine. On the other hand, there were people who believed that the nervous system is actually a reticular network. So there were two different uh, groups of people who believed in two different kinds of uh, we can say two opposite kinds of things. So Ramoni Kahal and others believed that neurons are individual entities or cells, whereas people like Held and Golgi, these people believed that the nervous system is a reticular network, the reticular or a reticular system. And the problem was that light microscopy was not advanced enough. The staining techniques were not advanced enough. But later, in 1950s, with the invention of the electron microscope, people were able to see that neurons are not a reticular network, but rather they are individual entities, and the interaction between one neuron and other happens through the presence of synapses, right? This was shown by a electron microscopy. So we had discussed in the previous week lecture, previous week that Sanford Paley and George Ballard, for example, they studied the structure of the synapse. They showed that there are like the neurons have synapses that are and the what are the internal structures that are present in the neurons and so on, right? So the use of the advent of electron microscopy was something that strongly supported the neuron doctrine, and this happened in the 1950s, again the golden decade of neuroscience. Next question: where did researchers discover action potentials are most likely to be generated in a neuron? So uh, this we had discussed briefly some time back, right? With regard to one of the previous questions, that people initially believe, like the people who were studying the action potential, for example, Jack Eccles and so on, people who are studying these things at that time, they initially believed that the action potentials are probably being generated in the dendrites or the cell bodies. That is what they thought, because they believed that the dendrites are just passive elements, and the dendrites and cell bodies just pass along the impulses that are being generated at the postsynapse. They just passively pass on this information to the, I mean, they do not play any role in any, they do not help in any integration of the impulses and so on. So people at the time were thinking that probably the action potential is being initiated somewhere in the dendrites or in the cell bodies in response to the uh, information that is coming from the presynapse. But to their surprise, they discovered that the action potentials are most likely to be generated at the axon hillock, or basically the axon initiation segment. So axon hillock is just another term for axon initiation segment. And later they understood that, okay, the dendrites are actually active elements. The dendrites and cell bodies actually integrate the information that is received from several synapsing neurons, and then based on the strength of the information and the nature of the information and so on, the action potential is finally generated at the axon hillock. Next question. What was the key discovery about dendrites that changed the original understanding? We just discussed that. What the key discovery is that, so we know that dendrites are not completely inactive, right? The dendrites are able to integrate the information. So dendrites are not completely inactive. Second, dendrites can only receive signals. No, that is not true. Dendrites not just receive signals, but they also uh, uh, conduct action potentials. So this is false. Third, dendrites can also conduct action potentials and receive feedback. Yes, dendrites can conduct action potentials, and they also receive feedback from the axons. Like the feedback that is coming from the axons to the cell body to the dendrites. So they can both conduct action potentials and they can also receive feedback. Dendrites are only found in specific types of neurons. No, all neurons contain dendrites. Final question. This also we just discussed a little while ago. What unique term did H.T. Chang introduce to describe dendritic spines? So, the term that H.T. Chang introduced to describe dendritic spines is the term gemmules. So, he said that dendrites have these components called gemmules, and these gemmules constitute a mechanical barrier that prevents the synaptic knobs from interacting directly with the dendritic stems. And how do these gemmules do that? That is because these gemmules probably have, I mean, because of the extremely slender stalks of these gemmules, they have very high ohmic resistance, and because of that, they can attenuate the signal that is supposed to reach the dendritic stem. So with this, I come to an end to the previous year assignment questions. Let me go back to the slides and just give recap whatever we had, whatever was discussed in this week's video lectures. In case any of you has any questions or doubts, you can stop me at any point and we can discuss. Yes, Soumya, your answer is correct. To the cell body, yeah, from the dendrites to the cell body. Yeah, so the axon hillock is basically the axon, the initiation segment of the axon, and that is where the action potential is initiated. Yeah, so yeah. So during the time that carbs and fat were, carbs and fat at that time were working on the the motor neurons. So, they were working on the end plate potential in the neuromuscular junction, right? Carbs and fat were working on the end plate potential in the neuromuscular junction using the frog neuromuscular junction. At the same time, Eccles was, like I said, was the student of Charles Sherrington. So Eccles started working with the spinal motor neuron, and he wanted to understand what is happening in case of the central synapses. So Katz and Fat had already shown that at the neuromuscular junction there is end plate potential that is being generated. That means that the acetylcholine that is being released by the synaptic terminals, basically over here in the at the synapses that are forming at the neuromuscular junction, the acetylcholine that is being released by the presynaptic terminal is able to generate a muscle, the end plate potential in the muscle, and that gives rise to the muscle action potential. This is something they had shown. Eccles was very Eccles, like I said, was someone who strongly believed that electrical transmission is the key to transmission at the synapses. But then because carbs and fat had shown that there is end plate potential that is being generated in response to the release of neurotransmitters. Eccles's idea was disputed, at least in the neuromuscular junction. But Eccles did not lose hope. He wanted to believe that in the central synapses, at least, there must be electrical transmission that is happening. So he thought that, okay, in the neuromuscular junction, it is probably chemical neurotransmission, but in the central synapses, the basis of transmission is electrical. So he kept on working on this for a long time. He used the spinal cord, the sympathetic ganglia, to come up with. His observations on the electrical transmission in the spinal cord. I'm sorry. His observations from the sympathetic ganglia in the spinal cord. He believed that it is because of electrical transmission. So then what happened is, uh, Eccles was in Oxford, right? Eccles was he was working as a research assistant with Charles Sherrington after his PhD. So after his DPhil, he was working with Sherrington in his lab. And when Sherrington retired, uh, because Eccles was a very very outspoken character and very pugnacious and so on, uh, it was not preferred. The environment at Oxford was such that they did not want to appoint Eccles and give him a laboratory, so that the position went to somebody else. So after Sherrington retired, someone else was given the position at Oxford. Eccles was not considered. So in that case, Eccles, being an Australian, he returned to Australia. He returned to Australia and he was working in a very small institute where was hardly any facilities, and it was very obscure, and he was almost going to fade into oblivion. But Eccles did not give up. He collab he still he also made the observation. So no, it was not just so he was also, I mean, Eccles was also one of the people, Eccles and Connor, right? Eccles was also one of the people who had made the observation about the end plate potential. So not just, but he still didn't want to give up on the idea of electrical transmission in the central synapses. So then what he did was he moved to New Zealand. So in New Zealand, he collaborated. He went to New Zealand. There was a research institution. It was a little better than his previous institution in Australia, but he had a lot of teaching responsibility. So he had to teach physiology, and like almost 500 hours worth of teaching he did in his first year there in New Zealand, Dunedin. So when he went there at University of Otago, he put together a group of people. He did not go alone. He worked with Archibald McIntyre, who was Wilfred Ral's supervisor, and he collaborated with several other people, and he wanted to understand what happens in the central synapses. He did not give up, basically. And Archibald McIntyre, when he had gone on a visit, he came back with several high-end equipments. I mean, high-end at that time, that were made by the famous German engineer. So Jan Friedrich he was the one who had also made he was one who also, if you remember the Hodgkin Huxley lectures, he was the one who had built these CROs and amplifiers and so on, right? So when Archibald McIntyre returned to New Zealand from his visit, he brought these CROs and amplifiers that were made by Jan. So, McIntyre was a very mild-mannered person, and Eccles just took over these high-end equipments that Archibald had, because Eccles himself did not have these. Eccles himself was still working with more old school equipments. He did not have access to these more modern equipment. But Archibald McIntyre, who was another new faculty there, McIntyre being relatively mild mannered, Eccles just took over these equipments and started using them along with his students and other collaborators. So Eccles continued to pursue this electrical transmission theory. I mean, his idea. And we know that the famous soup versus sparks debate with H. Dale and so on, right? Then very, like, this is something that we should really take inspiration from. The fact that, I mean, although Eccles was hell bent on his idea of electrical transmission, he was also the one who demolished his own hypothesis. So he did experiments, uh, like that he himself showed that the electrical hypothesis was wrong. So we'll come to that. So how did that happen? So when this chemical transmission, I mean the fact that chemical transmission happens in the neuromuscular junction came into the picture, and several other evidences came into the picture that it may not be an electrical transmission that is happening. Eccles was had become very depressed. He was not sure. I mean, he did not know what to do. At that time, Eccles was became friends with a famous philosopher who told him that, hey, why don't remember one thing, that there is never never a way to completely truly prove a hypothesis. Rather, it makes much more sense in science to come up with a hypothesis and then see whether you can disprove that hypothesis. The falsification or disproving of a hypothesis is much more better. I mean, makes much more sense, or um, is possible, rather than finding ways to prove a hypothesis, right? So there can always exist a way in which a hypothesis can be disproved, but there can never be, I mean, it is much more difficult, or it is much more improbable to prove a hypothesis. This was what Eccles was told by his philosopher friend. So, bearing that in mind, Eccles decided that he will generate a hypothesis of for the electrical transmission based on the again using his spinal using experiments involving the um, the transmission experiments involving again the central synapses, basically the spinal motor neuron related. So he said that, okay, let if I see that this hypothesis is getting disproved, the electrical hypothesis is getting disproved, then I will agree that it is indeed chemical neurotransmission that is taking place. So what he did was, he observed that when the sorry when the sensory nerves that are coming from the agonist muscles, if the agonist muscles are being activated, then the information that is coming from the sensory nerves and then goes to the spinal cord, and then finally the information goes to the motor nerves and finally goes to the muscles, right? So he saw that when an agonist muscle is being activated, then there is excitatory postsynaptic potentials that are being formed, or EPSPs that are being formed. When an antagonist muscle is being activated, in that case, there are so if an antagonist muscle is being activated, then the information that goes on, from the sensory nerve to the spinal cord to the motor nerve and so on. So in the motor nerve, it was being observed that there are inhibitory postsynaptic potentials that are being generated. So if an agonist muscle is being activated, there are excitatory postsynaptic potentials being recorded. If there are if the antagonist muscle is being activated, then there are inhibitory postsynaptic potentials that are being generated. Now, excitatory postsynaptic potential can be explained, right? Because when the agonist muscle is being activated, the excitation is happen in excitatory information is getting transmitted. But how do you explain inhibitory postsynaptic potential by using the electrical transmission hypothesis? Because electrical transmission hypothesis means that electric currents will flow, right? But inhibitory postsynaptic potential means that there is hyperpolarization of the postsynapse that is happening. It is not, the electrical currents are not flowing, rather, there is hyperpolarization that is happening. So, in that case, that means that electrical transmission is not the way by which the information is being passed at the synapse, because if there is electrical transmission of information, then the current should flow. There should not be an inhibitory postsynaptic potential that is being generated, but they are observing that there is inhibitory postsynaptic potential that is being generated. So that demolished Eccles's electrical hypothesis. Eccles himself conceded that no, electrical transmission does not happen in the central. It is not electrical transmission that is happening in the central synapse. Rather, there is chemical transmission that is happening, because of which inhibitory postsynaptic potentials are being generated. Later, it was shown that what happens is that uh, when the antagonist muscle is being activated, what happens is that the there is hyperpolarization arising because of increased conductance of potassium, and later it was shown that not just potassium but also chloride ions. So because of increased conductance of potassium and chloride ions, there is hyperpolarization that is happening, and that gives rise to the inhibitory postsynaptic potentials. And again, this happens through chemical transmission, basically through chemical neurotransmitters. Then Eccles went on. So this was all when Eccles was in Germany. Then Eccles received an offer to join a position at Australia. I think Australia National University. I please go back and check the exact. I forget the exact name of the university, but it was being newly constructed back in Australia, and Eccles got an opportunity to join there. So he went back from New Zealand to Australia. But at that time, it took almost 15 months for it was supposed to take almost 15 months for the new lab to be constructed. But for any other normal person, this would have become a very big deterrent, and it would have, because at that time was very critical. People were all in a race to do the experiments and so on. But for Eccles managed that time wonderfully. In fact, he spent 5 months traveling to America and Europe and so on. And he kept himself up to date with the Hodgkin-Huxley model, the finding that the observations of Katz and his colleagues and so on, and he synthesized all of that into his own book called The Neurophysiological Basis of Mind: The Principles of Neurophysiology. And this work that Eccles published, I think he did this in 1953, if I'm not wrong. So this that he published was it actually launched modern neurophysiology in a way, because he synthesized all of these different work of that time of neurophysiology into a book. This also we have discussed briefly about Ladislav Toc, who was a French person. And one thing I really wanted to discuss, which was also discussed in the video lectures, was that much of Ladislav's work went unnoticed initially because most of his studies were published in the French language. And like we discussed, the rest of the world, because they were all, the English was, and even now very strongly is the language of choice for uh, communications of the scientific communication. So Ladislav's work, which was published in French, or most of this work, that went unnoticed initially, although later some of his work, I mean not just later, at the time also some people did take note, but still majority of his work initially went unnoticed. Yeah, so one of the important contributions of Ladislav, other than all of these, is also the fact that he pioneered the use of aplysia as a crucial model system, invertebrate model system, and the fact that by listening to his lecture, Eric Kandel he requested Ladislav Locke that he wished, sorry, Talk to train him with this model. And later, Candle used aplysia to study study learning and activity dependent changes in learning and memory. So this is also something we had discussed during the course of the assignment questions, that the stretch receptor cells in crayfish. These were initially the use of these and study of these was initially started by J.S. Alexandrovich, but later, the Stephen Kuffler, especially, and Carlos, who was a postdoc, uh, these two people showed that showed using the stretch receptor cell, that there is depolarizing potential that gets that arises because of the stretch of the muscle spindle, and which causes the initiation of action potential and transmission of this action potential to the nervous ganglion. And one more important point is that this the Stephen Kuffler, this crayfish, the stretch receptor cells were also used by Stephen Kuffler and his laboratory to show that the dendrites indeed dendrites are indeed not just passive elements, they are also involved in the transmission of. Is any do you have any question, Soumya?
ma'am.
Okay. Yeah. Um, yeah. So, by using the stretch receptor cell model of Stephen Kuffler and his laboratory showed that depending upon the strength of the stretch, basically depending upon how much the muscle stretch is happening, the depolarizing potential that is generated, that is proportional to the muscle stretch that is happening. Basically, showing that dendrites to play a role, dendrites in the cell body to play a role in integrating the sensory information. Basically, they do play a role in integrating the receptor potentials that are being uh, generated with the action potential. I hope you have understood so far. Uh, Next, some of the other studies of sensory responses were initially were carried out by Edgar Adrian and Zotterman. This was again using extracellular recordings. So they managed to dissect up to single fibers and perform what was called extracellular single unit recordings. But the only limitation of these record, I mean, some of the limitations of these recordings were that the amplitudes of the recorded potentials were were very small, and because these were extracellular recordings, they reflected not individual cells but rather the combined activity of several cell populations, and because there was an overlap. I mean, it was a combined activity rather than that of individual cells. It provided limited insight into the actual membrane properties and the mechanisms that were actually taking place. And but this, remember, was in the early 1926, for instance, and it was in the 1940s and 50s when the intracellular recording started, right? This also we discussed. Even now, electroretinogram, in fact, is used for electrophysiological recordings, for example, from Drosophila retina, and so on. And it is also even used in clinical settings for certain disorders involving the retina. I would strongly encourage you all to go through these lecture video, through these research articles, so that you can understand. I mean, so that you can understand uh, the insights that these authors made. I mean, the insights that these authors had by just uh, performing experiments at the tissue level or the organ level, and the using equipments that are considered crude by current standards. Even using those kind of equipment, they were, and those kind of tissues and preparations, these remarkable scientists were able to make such brilliant observations that scientists of the modern day may not have been able to, would not have, would probably not have been able to make. So, Let me also recap Cahal's law of dynamic polarization and why it required revision. So, Ramon Cajal gave this law of dynamic polarization in the late 19th century, right? At that point, Cajal and others did not know that dendrites also have active properties. So he proposed that the information is just passively passing through the dendrites and cell bodies and it reaches the axon. But later, when the active properties of dendrites were uncovered, and there was extremely overwhelming evidence that dendrites do have active properties, Ramon Cajal's law of dynamic polarization did require revision because dendrites are not passive elements. And now, right, uh, now we know that dendrites do have voltage gated ion channels and they are capable of uh, uh, transmitting information not just passively but also they're capable of initiating action potential and so on. So far, do you have any questions? Does anyone have any doubts?
No, ma'am.
Okay. This we have already discussed in much detail regarding the uh, population spike in the hippocampus and the idea that hippocampal slices can be used as models for investigating trisynaptic circuits and long-term potentiation. And even now, people use mice hippocampal slices for studying phenomena like long-term potentiation and hippocampus. Although initially, I mean, it with this set of experiments, hippocampus emerged as a model system for these kind of experiments. Currently, it is a very popular model, especially the mammalian, especially mice. Hippocampal slices are commonly used for these kind of experiments. Yeah, so in the 1990s, when the patch clamp method was developed, that is when it was revealed that there are voltage dependent ion channels even in the dendrites. And the fact that the active responses of the dendrites are dependent on several factors. These include the geometry of the dendritic branches, the density of the channels in in the dendrites, as well as the what kind of neighboring channels are present. All of these are together responsible for the properties of the dendrites. And here I would also like to highlight the contribution of Wilfrid Roll. So far, all of the active properties in all of the when we had discussed the the work of Hodgkin and Huxley and the GHK equation and so on, we from that we realized that it is important that there is some kind of quantitative framework that can help us understand the biophysical properties of neuronal membranes, right? So that was something that was lacking for a while in terms of dendrites. So Wilfrid Roll was the one who proposed proposed the fact that proposed a model to understand how dendritic trees work, and this was the dendritic dominance model that he proposed. So uh, he said that the cell body and the dendritic tree can be modeled in terms of an equivalent cylinder concept. Basically, that the dendritic tree can be modeled as a cylinder to which the cell body is interact with which the cell body is interacting. And like we discussed, this was initially strongly rejected and it took quite a while to be accepted. And that happened much later. And Ral's biophysical studies contributed a lot to our understanding of the morphology of the dendritic tree and the properties of the dendrites and so on. So it was very important in reigniting the interest that regarding the properties of the dendrites. So these are the dendritic spines that we were talking about. Uh, and we had also briefly discussed about H.T. Chang's pioneering studies. So H.T. Chang was a Chinese who studied I mean who went all the way from China to America, and he applied to Fulton, and he went to Yale, and it was again something that was marked with a lot of struggles. And we discussed about Chang's observation on the of the dendritic spines, and he called them as gemmules. And he also functionally analyzed these dendritic spines in different kinds of neurons. So with this, I think we can end the class if no one has any doubts. Does anyone have any doubts? Synaptic knobs are basically the presynaptic terminal. At that point, he called them as synaptic knobs, but they basically mean the same as the presynaptic terminal. Okay. Uh, in that case, if no one has any further questions, I will end the session. Thank you very much for joining.
Thank you, ma'am.